Hello, Intro to Micro for Health Science students. This is your lecture that was supposed to be given on Monday, December 3rd, 2018. So just a reminder, we're not meeting in class today. There will be some questions posted online. Actually, what I've decided to do is everybody gets 4 points and you don't have to answer the questions online. I'll send out a message about that. The questions that I would normally ask that you can use for review for the exam, which is next Monday, will be posted at the very end of the lecture notes for this particular class. So take a look at the lecture notes after you're done listening to the recording and look at those questions, use those as review. Okay, the reminder, the final exam is Monday, December 10th from 10 to 12:45. In RH 101, this will be the typical time for the exam. If there is a change to this, I will let you know and tell you how we will proceed. We will not cover anatomy, as I've said before. I present some anatomy at the beginning of each section, just so you get an idea of where these diseases are located. I want you to focus on the microbes, their virulence factors, the diseases they cause, their symptoms, and their treatment, etc. So focus on those when you're studying. We're actually going to finish off where we left off on last lecture, which is tuberculosis. And as I said, there's no way I could cover tuberculosis in a minute and a half because there's so much information about tuberculosis, and this is a fascinating disease. If I don't give you enough information here, I would encourage you to go to the CDC and look up that information or read the articles that I'll be posting in here. So tuberculosis is a very big problem in Alaska and has been for a really long time. At one point in Alaska, up to 3/4 of all kids were infected with tuberculosis. Those kids were sent to sanitariums, which I'm showing on the right here, and that was very disruptive for Alaska Natives and Alaskans living in Alaska at that time, because the children were separated from the rest of their family. They weren't schooled in these sanitariums, they weren't given an education, and this completely disrupted their family structures and their education. Alaska still has very high rates of tuberculosis, as seen in this article, and we have the second, in 2016, we had the second highest rate in the nation. Tuberculosis is not just a problem in Alaska, though, obviously. It's a problem across the world. And this is a map, an incidence map from 2010, showing that in the very darkest colors, there are more than 300 cases per 100,000 people. And even here in the United States, you can see that we have that light orange color, which is indicating more than 100 cases per 100,000 people. That's a pretty high rate for an industrialized country. If you'd like to know more about this, the World Health Organization keeps track of this information, and you can find more information on their website. And I would like to stress down here, tuberculosis is the leading microbial disease killer in the world. And here's a nice article talking about how tuberculosis has affected Alaska specifically, both the history and current treatment. And we're going to get into treatment in just a minute. In Alaska, we spend millions of dollars trying to make sure that people take all of the pills that are in their regimen, which can last for up to 9 months, because rates of resistance, antibiotic resistance, are so high, and because this is a disease that can be transmitted from one person to another. And we don't want another huge outbreak in Alaska. So how does this disease progress? It is inhaled in water droplets. The bacteria that causes this is Mycobacterium. It infects the respiratory tract, usually via inhalation from infected individuals. The bacteria lodges deep in the alveoli of the lungs. So if you've forgotten where the alveoli are, they are really deep in the lungs. They're basically the little air bubbles at the little bubbles that collect air from oxygen being breathed in at the very end of the lungs. And remember that this is highly vascularized and that there are alveolar macrophages present in the alveoli. And in fact, tuberculosis takes advantage of that and infects those macrophages, as you can see here. So the bacteria, Mycobacterium, are ingested by, are actually phagocytized by macrophages, and then the bacteria are able to survive in those macrophages. The bacteria replicate freely within the macrophages, eventually killing the phagocytes, and the bacteria are then released from dead macrophages and are phagocytized by more macrophages, which allows the bacteria to replicate once again. As these macrophages are dying, as the macrophages are phagocytizing the bacteria, they present the antigens from the bacteria on their surface. Is this going to be on MHC class 1 or MHC class 2 molecules? Most likely it's going to be on MHC class 2 molecules because these are phagocytes and they present bits of microbes via the endocytic, or via the vesicles, the phagocytic vesicles that were used to bring in the microbes. So these would be presented on MHC class 2 molecules. So the antigen is presented on the surface of the antigen presenting cell, and that is presented to T cells, which are then recruited to the site of the infection in the alveoli. These T lymphocytes then produce lymphokines that attract and activate more macrophages and trigger inflammation. So now all of a sudden you have inflammation in the lungs as well, deep in the lungs. And these macrophages become tightly packed in these things called tubercles. And these tubercles have live bacteria in the very center, and then they're surrounded by macrophages, and this forms within a 2 to 3 month period. I've mentioned before that Mycobacterium is a very slow replicating bacteria. It takes up to 28 days to form a colony on a plate, and so these bacteria are replicating inside of the macrophage, and it takes a while for them to do that. And as they're replicating, they're killing the macrophages, recruiting more macrophages and T cells, and you have these nice tubercles formed. Other cells deposit collagen fibers, enclosing infected macrophages and long cells within the tubercle. Infected cells in the center die, releasing Mycobacterium tuberculosis and producing caseous necrosis. So you remember from our earlier lectures that necrosis means death. We saw that in necrotizing fasciitis. So these are dead cells that eventually create these granulomas. The death of the tissue takes on a cheese-like consistency, and that is, and these are formed within the alveoli. Eventually, tuberculosis can break out of these tubercles, these caseating granulomas. They can burst out and then disseminate throughout the body, and Mycobacterium tuberculosis can then form infections in other parts of the body like the lymph nodes, bone marrow, spleen, kidney, spinal cord, and occasionally, rarely, in the brain. Now, I forgot to mention on the previous page and on this page, cord factor. Cord factor is one of the major virulence factors of tuberculosis. So on this page, cord factor protects tuberculosis from killing by macrophages. And on here, cord factor also helps create caseating granulomas. So this is an important virulence factor that's found in Mycobacterium tuberculosis. So what does this look like in an infected person? So here we see an X-ray. This is how it's typically diagnosed. And you can see that there's a large area of infected alveoli over here. We also do a skin test. Many of you have probably had this, or your kids have had this as they go through school. A little bit of tuberculin is injected underneath the skin, and as we talked about in our lecture on hypersensitivities, the tuberculin results in a what? Type 4, a type 4 hypersensitivity. And this is due to the T cells reacting to the antigen. And so you get a red bumpy area. Now you can also get this if you've had the vaccine. So if somebody has been vaccinated, they need to tell the healthcare workers because this will also result in a red inflamed area. And the red inflamed area is not 100% diagnostic, and so typically if the reaction is negative, then people are retested 1 to 3 weeks later, and that's because if a person is newly infected, it can take a while for that T cell response to become activated. And so if you've just been infected, you may not get the red bump initially when given the tuberculin test. You can also do a sputum test, and for this, sputum is collected and an acid-fast test, acid, A-C-I-D, fast, F-A-S-T, an acid-fast stain is done on the cells. And what you look for are these red rods seen here. Those are the Mycobacterium tuberculosis, and they stain red. The treatment for tuberculosis takes a long time. It's up to 9 months of treatment using a combination of different antimicrobials. So here we see isoniazid, rifampin, pyrazinamide, and ethambutol or streptomycin. And the reason for so many different antibiotics is because this can develop antibiotic resistance fairly easily. And in fact, many of the strains found worldwide now are antibiotic resistant. And in addition to that, Mycobacterium tuberculosis can develop antibiotic resistance in the body during the treatment. And so people, while they're taking these antibiotics, will be monitored for resistance, bacteria gaining resistance to these different antibiotics. Because the treatment is so long, patients are also given directly observed treatment, short course, DOTS, and that is exactly what it sounds like. Patients are watched as they're taking the medication to ensure that they take all of the pills. There's a nice, in the ADN story that I showed earlier, there's a nice description of that. And I'm showing this map, which is from the World Health Organization, to show the multi-drug resistant strains that are found throughout the world. You can see that here in the United States, we have an incidence of about 3%, up to 3% of our cases of tuberculosis are multi-drug resistant. In other places, like Russia, seen here, there are rates of up to 18% drug-resistant, multi-drug resistant strains of tuberculosis. So that's it for tuberculosis. There's obviously a lot more you can learn about tuberculosis. If you'd like to, go to the CDC and World Health Organization or read the section in this particular book. It's a fascinating disease, and it affects not only Alaska in the United States, but there are a high number of cases of tuberculosis worldwide. So now we're going to move on to pertussis. Pertussis is a disease that infects the respiratory system, obviously. It produces a very characteristic cough in young children who are infected with with pertussis. There are 3 different stages of the symptoms. In stage 1, that's the Catterall stage, and the symptoms look an awful lot like a cold. So runny nose, low-grade fever, mild occasional cough. At this point, though, the patient is highly contagious. So you think about when you have kids playing with other kids, if they have a cold, kids are usually allowed to play with other kids. But if they develop symptoms that are really characteristic of whooping cough, which is what pertussis is, then at that point, they're actually in stage 2, the paroxysmal stage. And at that point, they have fits of numerous rapid coughs with that characteristic whooping sound. And in that case, in this case, you can also see vomiting. Children may also exhibit vomiting and exhaustion after coughing fits, in part because this is an infection that affects the lungs, and it's hard for the patients to get enough air to feel oxygenated. And then stage 3 is the convalescent stage, lasts about 2 to 3 weeks. And in this particular part of the infection, patients are susceptible to many other respiratory infections, and recovery is gradual. So I wanted to show a video of babies with whooping cough. I'm just going to show the first one, and I'm only going to show a few seconds of that first one, and you can watch the entire videos using the links below. So you can see this is a persistent cough, and the baby just never really seems to have enough time to get enough air. And the cough just gets worse and worse. In some cases, the baby can turn blue because they're just not getting enough air. It's very scary. The baby stopped breathing there for a little bit. You can hear that. So that was just one example. You can watch the rest of that video in the links. It'll be in the lecture notes. So here's a summary. I'm not going to go into greater detail on pertussis, except to say that it has a virulence factor that allows the bacteria to produce a toxin called pertussis toxin that affects these cilia that are on the epithelial lining of the trachea. Those cilia are no longer able to move mucus up and away from the lungs, and that allows the bacteria to get into the lungs and just kind of hang out there. This is caused by Bordetella pertussis, which is a gram-negative coccobacillus. So think about what shape a coccobacillus is. And the virulence factors include adhesins that allow the cells to attach to tracheal cells, that allow to other virulence factors that allow the bacteria to survive within the phagosome, and then, as I said, pertussis toxin, which causes human cells to make more receptors for Bordetella and can damage those cilia. Signs and symptoms we've gone over. Incubation period is 6 to 20 days with an average of 7 to 10 days. Susceptible kids are unimmunized children. This is much more damaging in children than it is in adults. Treatment is supportive care, and erythromycin has only little effect on the course of the disease, but helps eliminate it in the respiratory tract, and then thus helps reduce the spread of pertussis to other kids. And of course, the prevention is a vaccine. Okay, so the last thing we're going to talk about for the respiratory system is mycosis in the lower respiratory system. So in this particular case, it's really important to get a history of a patient's history of where they've been traveling. And that's because there are many different types of yeast that can cause diseases in the respiratory system, but they tend to be localized in very specific areas. So for example, if you have a patient that has a characteristic prolonged cough that doesn't seem to be bacterial or viral in origin, and you suspect that it's fungal, and you know that they've been traveling along the Mississippi River, then you would assume that they may have blastomycosis. Whereas if your patient has been traveling to parts of Africa or parts of South America, as you can see here, your first thought might be histoplasmosis. And then there's coccidiomycosis, which is found much more prevalently along the southern border of the United States and sporadically or in various places in South America as well. So histoplasmosis is the one that we're going to focus on. This is a fungi that can turn into a yeast form when it's inhaled, and it reaches that much warmer body temperature of 37 degrees. It converts from a fungi form. It converts into a yeast form. And so here you can see that this originated in the environment, most often in the feces from different types of animals. Shown here are bats. This is inhaled as spores, and then once it gets into the lung, it converts into yeast that are covered in capsules. And of course, that prevents the fungi from being phagocytized by macrophages. It spreads to the lymph nodes and to the blood. And here you can see these yeast replicating inside of macrophages. And so they can spread there to other parts of the body. And this becomes an issue, especially in immunocompromised patients. Most patients, 95% of all patients who develop histoplasmosis will recover very quickly, but immunocompromised patients can develop disseminated infections due to the yeast replicating in those macrophages. Unfortunately, the symptoms may mimic tuberculosis, which is much more frequent, is in a much higher incidence in most patients. And so the first thing that doctors usually think of is tuberculosis rather than histoplasmosis, and that can delay the treatment. And in immunocompromised patients, that may result in the bacteria disseminating to the spleen and the liver, causing an enlarged spleen and liver, which eventually can cause death. So this is what the fungi look like when they're replicating inside of macrophages, and this is how they become disseminated throughout the body. They can also form calcified nodules in the lungs. These are the calcified nodules in the lungs of a relatively healthy patient who is asymptomatic. And so even though all of these calcified nodules are in the lungs, they may not have prolonged symptoms. Disseminated histoplasmosis in an AIDS patient can look like this, where you have huge red bumps all over the patient's body. 95% of patients recover quickly. Immunocompromised patients have the fungi can spread through infected macrophages to the rest of the body, leading eventually to death. Symptoms can mimic tuberculosis, as I said, and it's treated with antifungal meds. You have to treat for up to a year because the fungi can get into different parts of the body. This is just a summary. It'll be in the notes, and you can take a look at this at your leisure. I like talking about histoplasmosis in part because it's one of the reasons that we tend to think of tombs and other areas as being cursed, and that's because people would venture into places where bats had been hanging out for a long time. Histoplasmosis would be caused when people would inhale the fecal material of these bats in these contaminated areas. And where do bats like hanging out? Nice dark places like tombs. And so the idea of the cursed tomb comes from histoplasma. Okay, so that's it for respiratory infections. Now we're going to talk about diseases of the digestive system. I'm going to talk about the digestive system in general, then talk about dental diseases, mumps, peptic ulcers, gastroenteritis, hepatitis, and I'll finish with parasitic diseases. So let's start with an overview of the digestive system. The digestive system includes everything from the mouth all the way to the anus. So anything that you can take into your body and by eating, and that can go through the digestive tract can potentially lead to infections. Here you can see that the digestive system includes the mouth. So those of you who are thinking about going into dentistry or who will be doing predental classes, there are many diseases that can affect the mouth. We're only going to talk about a couple today. And then the digestive system includes the esophagus, which is the tube that leads from the mouth down to the stomach. Food can go through the stomach and then enters into the small intestine and large intestine and is released from the anus. Included as part of the digestive system, which most people don't think about, is the liver and the gallbladder, and the pancreas. And that's because these are all things that can be affected by or can affect the digestion of food. So the gut is basically a big tube that has a whole bunch of ridges inside of it. And those ridges are called villi, and they increase the surface area of the gut. That leads to more surface area for nutrients to be absorbed by the epithelial cells that line all of these villi. And that's a good thing, because with all of that surface area, we can absorb a lot of nutrients from food that goes through our digestive system. These nutrients can also be absorbed from not just directly from our food, but as byproducts from the foods that is digested by our microbes. So this is showing the intestinal barrier to pathogens. So microbes can come in through the mouth and enter into the stomach and into the gastrointestinal tract or the GI tract, the intestine. The bacteria in our intestine help prevent infections by just colonizing the intestine and also producing antimicrobial factors. These bacteria also stimulate the immune system, so that's what we see here. We see IgA that's being released into the mucosal area of the intestine, and that's due to stimulation by our beneficial microbes. There's also a nice thick mucus layer, and of course mucus prevents pathogens from coming closer to the intestinal layer. The intestinal layer epithelial cells are seen here, and those are what are absorbing nutrients from the food that's being digested in the intestine. And then there are a number of other immune factors like Paneth cells and just a large number of immune cells that are constantly in the intestine, and those immune cells are being stimulated by beneficial microbes so that they are ready to attack pathogens as they come into the intestine. The microbiota includes bacteria and fungi and protozoa and viruses, so it's really diverse. There are hundreds of species of bacteria alone, possibly thousands of different types of viruses, an awful lot of fungi and protozoa in there. Most of the bacteria are gram-negative bacteria, including Bacteroides and Enterobacteriaceae. The gram-positive bacteria that are found in the gut tend to be Lactobacillus or related microbes. Okay, we're going to start with diseases of the mouth. So here we have the tooth structure of the mouth. And I'm starting with this because we're going to talk about diseases of the teeth. So the tooth is basically enamel that covers dentin, which covers pulp. The pulp is highly vascularized. You can see the red and the blue here are the blood vessels, but there's also an awful lot of nerves in here. And that's why if you get a hole in that enamel, it hurts because those nerves are stimulated. Underneath the teeth are some really nice blood vessels and nerves. And again, you'll probably recognize that from the times that you've flossed your teeth and blood has appeared, and that's because there's a lot of blood underneath in the gums. Now this enamel is the hardest surface in the body, and that's because it has almost no water and is made almost entirely of minerals. Dentin is softer, so cavities that are able to form holes in the enamel and get into the dentin spread through the dentin faster than the enamel. And I like this picture because it's also showing bacteria. We have normal microbiota in our mouth, and what's interesting is that the microbiota between this tooth and this tooth may be different than the microbiota between this tooth and this tooth. And they protect just as microbiota in other parts of the body. Now, what happens when we get a tooth infection? This is often referred to as dental caries. And if it affects the gingia, gums, it's called gingivitis. Again, we have itis, so that means inflammation of the gingia here. So here we have dental caries, which is basically cavities. Bacteria colonize on the surface of the tooth enamel, and this produces a biofilm, which allows other microbes to come in. Now, what's interesting is that these bacteria are not producing toxins. They may be producing toxins, but that's not what damages the enamel. What damages the enamel is the acidic environment that these bacteria produce. So the acid from fermentation of food destroys the enamel. The treatment is just to fill in the cavity, and that should prevent the bacteria from causing a bigger hole. But if that hole should form in the enamel, now we have a cavity, and if it's allowed to spread, then it gets deeper into the dentin and gets into the area where the nerves are, and it can spread from tooth to tooth. You can also get gingivitis, which is the swelling of the gums. It's often found in mouths that have a disrupted microbiota. And this is also referred to as periodontal disease. Microbes that can cause this include Piriformis gingivalis and Streptococcus mutans. In this case, there are proteases that break down the gingival tissue. So in the case of cavities, it was acid produced by microbes. In the case of periodontal disease or gingivitis, it's proteases that activate inflammation in the gums. This can lead to swelling and bleeding and tenderness and redness. And if you have extreme gingivitis, then you have something called trench mouth, which can also be referred to as acute necrotizing ulcerative gingivitis. The treatment is scaling and antibacterial rinses. The mineral on the teeth is irreplaceable, and the gums, it's not irreplaceable. Irreversible solubilization of tooth mineral by acid produced by bacteria in dental plaque can produce the cavities. Acid produced by bacteria digesting fermentable foods like carbohydrates and sugar results in loss of mineral from the tooth. So this is the disease at a glance. Take a look at this in your notes later. Now we're going to move on to disease of the salivary gland. So we're remaining in the mouth, and we're going to talk about mumps first. Actually, we're only going to talk about mumps. So the incidence of mumps has been increasing as people are decreasing vaccination of kids. So here we have the mumps in the cases of mumps in the United States by year. You can see 2000 to 2005, there were very few cases. We had a spike in 2006 when people stopped vaccinating kids. And then we saw a decrease, increase, decrease, increase. Here we are in 2018. We've had a few outbreaks of mumps in the United States this year. This is not an updated graph. I assume that by the end of the year, this will be closer to 2017. We've had a number of outbreaks in Anchorage as well. So there was a big outbreak of mumps in, I believe, March of 2018. So just this past year, we had a large number of mumps here in Alaska. And that's indicated here as well in this map, where Alaska is a dark red color due to the number of cases of mumps in that particular outbreak. So what is mumps? Mumps is an infection of the salivary glands. There are actually a number of different infections that can result in mump-like symptoms, so swelling of the salivary glands. In this particular case, the virus enters via the respiratory tract. Mumps virus causes mumps. It's an enveloped, helicose, unsegmented, negative single-stranded RNA virus, and this can enter into the body via the respiratory tract, invade the blood, and disseminate. It can cause, rarely, inflammation of testes, meninges, pancreatitis, and deafness. In the case of mumps, 2 doses are required to be effective against mumps, and 2 doses is only 88% effective, which means that adults in outbreak areas typically require a 3rd dose just to boost the immune system against the mumps virus. And here you can see that this young adult male has mumps, so it's found not just in children, but it can be found in adults as well. So that's it. That's all I'm covering for mumps, because this is a vaccinated disease that we can treat with vaccinations. So again, get your kids vaccinated. Okay, So now we're going to move on to diseases of the stomach. We're going to start by with peptic ulcer disease. The first one we're going to start with is Helicobacter pylori. This is a gram-negative spirochete. So here you can see the gram-negative bacteria, Helicobacter pylori. It has flagella and adhesins that allow the bacteria to adhere to the cells that line the stomach. It also has a protein that inhibits acid production by stomach cells, which allows it to create an environment that allows the bacteria to grow. The stomach has a very low pH, and most bacteria do not survive that very low pH, but Helicobacter can prevent the stomach from producing that acid, which makes the environment more neutral and allows the bacteria to survive. It also creates urease, which neutralizes stomach acid, and it has enzymes that inhibit phagocytic killing. The bacteria get in through are ingested, they neutralize the stomach acid, and they get through the mucus layer by use of their flagella, which helps them get deep through that mucus layer and get closer to the epithelial cells that line the stomach. There they produce toxins and create inflammation that cause the layer of mucus to become thin, which allows more bacteria to get in and also allows acid from the stomach to get close to the epithelial cells. That damages the epithelial cells in the stomach and leads to an ulcer and allows the bacteria to get even deeper into the stomach lining. This can eventually lead to cancer with the bacteria replicating deeper in those tissues, and it also leads to ulcers. Risk factors for Helicobacter pylori include living in crowded conditions with no reliable supply of clean water, because this is a microbe that infects by ingesting contaminated food or water. Or you can drink the bacteria, as this professor did. Dr. Marshall wanted to prove that Helicobacter could cause ulcers, and so he drank a flask of bacteria. I don't recommend this. I don't recommend drinking the bacteria that you're studying, but it was effective. He developed ulcers as a result of drinking Helicobacter, the flask of Helicobacter, and that resulted in doctors around the world realizing that Helicobacter does indeed cause ulcers. It can also cause cancers, including cancers that are particularly high incidence here in Alaska, such as stomach and gastric cancer. This is relatively rare in the general population, but Alaska natives have a higher risk of gastric cancer due to Helicobacter pylori. And just want to throw in, Holly Martinson is a professor here at UAA who studies Helicobacter pylori and the connection between Helicobacter pylori and gastric cancer in Alaska natives. There is an argument for keeping Helicobacter around, and it's a pretty good argument, and it's called the hygiene hypothesis. And if you want to learn more about that, you can check out this link, and it'll be in the show notes. Not the show notes, the lecture notes, sorry. And here's a nice summary of Helicobacter. It can be treated with amoxicillin and clarithromycin, which is given in conjunction with acid-blocking drugs for 1 to 2 weeks. This can also disrupt the microbiota, and so this is a treatment, but it has to be, patients have to be monitored to make sure that there are no other effects as well. Prevention is good personal hygiene, adequate sanitation, proper food handling to decrease fecal oral transmission, and lifestyle changes to reduce risk, including dietary changes to reduce stomach acid imbalances, and reducing consumption of alcohol, tobacco, and aspirin-like pain medication that can affect the acid in the stomach. Okay, so that's it for Helicobacter. Now let's move to gastroenteritis. So we started with diseases that affect the mouth and the salivary glands, and then we talked about diseases of the stomach. Now we're going to talk about diseases of the the intestinal tract. These are diseases that can cause diarrhea, and diarrhea causes outbreaks worldwide. So here I'm showing a ProMedMail search for diarrhea over the last year, and you can see that there were at least 53 different updates on diarrhea outbreaks. Now, these are big problems that can lead to death due to nutrient loss and dehydration, and so they're monitored particularly closely in Asia and Africa and the Americas because this is such a big killer of kids and elderly folks around the world. The symptoms include nausea, vomiting, diarrhea, abdominal pain, cramps, and in the case of dysentery, loose, frequent stool containing mucus and blood. Now, this particular picture you might be familiar with if you've ever had a gastroenteritis infection, and this person looks pretty miserable, but I think that this more accurately reflects what it feels like when you have gastroenteritis. You feel completely fatigued and like you don't want to leave that toilet because you're gonna be either throwing up or have loose stools. Pretty miserable. Inflammation of the stomach or intestines is caused by bacteria. It's usually associated with contaminated food or water and poor living conditions because if you don't have running water, it's hard to wash your hands in between using the bathroom and preparing food, which is the main way that the bacteria is able to get in. We're going to start with viral gastroenteritis. There are 4 different types of viruses that cause most of the diarrheal infections caused by viruses. These include rotavirus, norovirus, adenovirus, and astrovirus. And you can see these representatives of these here. These are infections that affect people worldwide, but are much more, have a much higher incidence in countries that don't have access to good running water. Most cases of gastroenteritis are self-limiting, meaning that you don't have to treat them, and they appear within 12 to 24 hours and resolve within 60 hours, so just a couple of days. Antidiarrheal medication is not recommended, and that's because antidiarrheal medication slows the emptying of the intestinal tract, and what the body needs to do is get those viruses out of the gastrointestinal tract. And so you really want to keep that diarrhea going long enough to clear those viruses out of the system to prevent further symptoms. These are lytic viruses. So take a minute and think about the difference between lytic and lysogenic. They infect the intestinal epithelial lining of the intestine. Treatment is based on fluid or electrolyte replacement. So basically just replacing the fluids that are lost during that diarrheal infection. Prevention involves proper treatment of water and sewage and good hygiene practices. And there is a vaccine for rotavirus that's usually recommended for young kids because this can cause pretty serious infections in young kids. Now for bacterial infections of the gut, we're going to start with shigellosis. So shigellosis is caused by Shigella bacteria. And here we can see a Shigella bacteria attaching to the epithelial cell of the colon. This is the epithelial cell, here's the nucleus. This bacteria induces endocytosis. So epithelial cells typically don't endocytose bacteria, but Shigella induces endocytosis, and that allows the bacteria to come in the vesicle. And then the bacteria Shigella breaks out of that vesicle to replicate in the cytosol. Now it's able to take over the actin fibers of the host and use those to invade neighboring epithelial cells. This allows the bacteria to evade the immune system because it spreads from cell to cell without leaving the cell, and so the immune system doesn't recognize that these cells are infected. Eventually, an abscess forms when these bacteria replicate and kill the epithelial cells, and then the bacteria are able to get into the blood because the intestinal cells that line the intestine have there are a number of blood vessels just underneath the cells that line the intestine. And that's because those blood vessels are there to absorb the nutrients from the intestine and spread that throughout the body. But bacteria that are able to cross that intestinal barrier are able to get to those blood vessels and create septicemia. Shigella, though, is destroyed by phagocytes, so no septicemia is formed. This is really good for us because we're able to stop Shigella infections in the intestine. Shigella has a number of virulence factors, including adhesins that allow them to attach to the epithelial cells. They have pili and fibriae, which also allow them to attach. And here you can see the pili and fibriae attaching to host cell receptors. Symptoms include bloody diarrhea, and that's because of those lesions caused by dying epithelial cells that allow blood to get into the intestine. Diagnosis is to ID the bacteria in the stool, and treatment is to wait for it to pass. Another interesting virulence factor of Shigella is the type 3 secretion system that allows this bacteria to transmit proteins directly from the bacteria into the host cell. So it's basically a tube that forms between the bacteria and the host cell that directly injects things into the host cell. And then this is showing the Shiga toxin. Shiga toxin is an AB toxin. And that's an interesting toxin because the A part is the active part and the B is the binding part. So the B part of this toxin binds to the host cell and allows and triggers the host cell to take up the toxin. And then the B and the A part separate, and the A part is what actually activates, is active in the cell, and it inhibits protein synthesis. And when protein synthesis is inhibited, eventually the cell dies because those proteins are needed for regular metabolism in the cell. Okay, so that's Shigella. Produces Shiga toxin. The Shiga toxin is also found in E. coli, which is a bacteria that's closely related to Shigella. Now, Shigella and Salmonella are both gram-negative bacteria that are able to cause diarrhea in the infected host. And so what I'm going to do now is I'm going to show you the life cycle of Salmonella and talk about how it's similar and different from Shigella. So if you remember, Shigella was able to be endocytosed, and then the bacteria replicates in the cytosol. Salmonella attaches to epithelial cell linings in the small intestine, similar to, which is a different site than Shigella. So Salmonella binds to the epithelial cells in the small intestine, whereas Shigella is found in the colon, which is part of the large intestine. So Salmonella attaches to the epithelial cell lining of the small intestine, triggers endocytosis just like Shigella did, but then it multiplies within the vesicle. So this is different than Shigella. Salmonella replicates inside the vesicle. Shigella replicates inside the cytosol. Now this is interesting because it differentiates Salmonella from Shigella. Salmonella also kills the host cell, inducing fever, cramps, and diarrhea, and then can get into the blood. Now, unlike Shigella, which was killed by phagocytes, Salmonella is able to spread in the blood and cause septicemia. So there's another big difference. So Salmonella causes diarrhea and a rose-colored rash because it can get into the blood and weaken the blood vessels to cause that rash. It can also cause typhoid fever if you have the right strain, which can induce which causes intestinal hemorrhaging, kidney failure, and peritonitis. Now, salmonellosis is usually self-limiting, but typhoid fever can be much more. So a salmonella infection can be self-limiting unless you are infected with a bacteria that can cause typhimurium. typhi. So this is a table that contrasts Salmonella enterica serovar typhi versus Salmonella enterica serovar typhimurium. Now, typhimurium is self-limiting and is not normally fatal, whereas typhi causes typhoid fever and is often fatal. For typhi, a patient needs to be treated with antimicrobial drugs, whereas with typhimurium, it's self-limiting. So it's really important to not just identify the species of bacteria that's causing this infection, because these are both Salmonella enterica. So this is the genus, this is the species. But it's also, in this case, important to identify the serovar, whether the patient has typhi or typhimurium. There are other differences as well, including where the microbe is obtained from. For typhi, it's food or water, and for typhimurium, it's most often associated with chickens and eggs. Okay, so in the notes, you have this nice little chart to compare Shigella and Salmonella. You can fill that out as a nice little study tool. I wanted to mention just briefly, Salmonella enterica is one of the cases of bioterrorism here in the United States. So there have been a few cases of bioterrorism in the United States. The one most people are familiar with is the use of blankets to spread smallpox to infect Native Americans. But Salmonella enterica was also used as a bioterrorism weapon here in the United States. There's a fascinating story and podcast about this. Here's the podcast and here's the story of the case of a cult in Oregon, I believe it was, who spread Salmonella enterica on salad greens in restaurants to try to prevent people from voting. So it's a fascinating story. Check it out if you have time at some point. So here's a nice little summary of Salmonella and typhoid fever. Take a look at this as a good study tool. In the meantime, though, I'm going to move on to E. coli. I'm only going to talk about E. coli briefly. This is Escherichia coli, is a genus and species that has many different serovars, just like Salmonella, but it has many more serovars. Each of these different strains can cause a different type of infection, and they vary widely. So K12, for example, we use in the lab. It doesn't cause infections at all. But if you're infected with something like OH157, which is named based on the proteins on the surface of the bacteria, then you can have a very dramatic diarrheal infection that can often lead to death. The treatment is replacing lost fluids and electrolytes. And again, just like with the viruses, antidiarrheal drugs prolong the symptoms. Antibiotics can induce OH157H7 to increase Shiga-like toxin production. And this infection can also induce temporary lactose intolerance, which I just find fascinating. These are the virulence factors of E. coli. We're going to talk about this, not the virulence factors, the different strains of E. coli. We're going to talk about E. coli again when we get to diseases of the kidneys in the next lecture. And so I'm not going to talk about that much anymore. Okay, now we're going to move on to Vibrio cholera. Vibrio cholera causes a large number of outbreaks throughout the world, and it's typically associated with things like natural disasters. In those cases, people who are coming in to help people recovering from a natural disaster often bring Vibrio cholera with them, which is interesting. We used to think that the Vibrio cholera infections that came with natural disasters was due to bad hygiene and poor water and untreated water, but now we know it's due in part due to that, but it's also due to people coming in from other countries to help with the help people after the natural disaster. Vibrio cholera has this very characteristic comma shape to the bacteria. You can see that it's slightly curved. It has flagella that allows it to get close to the intestinal walls, the intestinal cells. Once they come in, this produces a toxin that can disrupt the adenylate cyclase and result in an increased secretion of chloride and sodium ions, and that results in water leaving the cell. That causes extremely watery diarrhea, so much so that patients who have cholera infections are often placed on these cots that have holes in the middle, and then the diarrhea is collected in these pots underneath, or these buckets, and you can see that this diarrhea is just watery. It's often referred to as rice water consistency. It looks like the water that comes off of boiling rice. The treatment is hydrotherapy. So this is a patient before hydrotherapy and after hydrotherapy, and that's basically just replacing the water that's lost during the infection, this part with the toxin. Now there is a vaccine that's being developed and it's now approved in the United States. This is a great article on the vaccine production for cholera and also the cases of cholera infections worldwide. So, in this particular part of the lecture, I've talked about a large number of bacteria that are all able to cause diarrhea. These are monitored by the CDC, and every couple of years a food safety report is released that shows how well we're controlling cases of these different bacteria that can cause gastrointestinal disease. Now, we didn't talk about Campylobacter today, but we did talk about E. coli OH157. In the past, we've talked about Listeria. We talked about Salmonella and Vibrio today, and in other lectures, we've talked about Yersinia. And so, as you can see, in 2015, this is the 2015 rate, and for some of these, we've had some increases in incidence. So, for example, Campylobacter has increased in the United States, but we've also seen some dramatic decreases. So, for example, we've increased our food safety procedures, and that has led to a big decrease in the number of cases of E. coli OH157 reported. So, mixture, good news, bad news. Now, these are all gram-negative rods that are causing these different diseases. This is a dichotomous key to differentiate between the different types of gram-negative bacteria that can cause intestinal diseases. So, for example, if you have a straight rod that's able to use lactose as a nutrient source and is citrate positive, you probably have Klebsiella, whereas if it's citrate negative, you most likely have E. coli. This is also a good way to differentiate between Salmonella and Shigella. As I said, they both look very similar, and E. coli, Salmonella, and Shigella are all very similar in shape and in other ways, and so, but they can all be have very different virulence factors, and so it's important to determine which one is infecting your patient. And so this is one way you can determine that. So this is a summary of the different types of bacteria that we've talked about. The only one on here that we haven't talked about is traveler's diarrhea. We talked about it actually in passing when we talked about E. coli. And then these are the other microbes that we talked about. We did not talk about C. diff. C. diff is Clostridium difficile. It's a part of the normal microbiota, but when the microbiota is disrupted, most often by antibiotic treatment, then Clostridium difficile is able to replicate at a much higher rate, and this can cause diarrhea. This can cause a long-term infection. In some cases, it can only be treated with a fecal transplant, which is fascinating all in of itself, and if you want to at a later time, you should look into fecal transplant advances. It's pretty fascinating. Okay, to finish off gastrointestinal diseases caused by bacteria, we're going to talk about Staph aureus again. Now, we've talked about Staph aureus infections on the skin, and we've also talked about Staph aureus infections in other parts of the body. Staph aureus can also cause diarrhea. The interesting thing about this is that it's not the actively replicating bacteria that causes diarrhea, it's actually the toxin production. So what happens is Staph aureus is able to grow on some sort of food that has been stored improperly. Typically, it's food that was left out at room temperature that should have been put in the refrigerator. As the bacteria replicates, it produces toxins, and those toxins are heat stable. So when the food is reheated, it doesn't kill the toxins, although it may kill the bacteria. Those toxins then cause, trigger muscle contractions, nausea, and may cause intense vomiting, and can also cause diarrhea. So we're showing that here. These outbreaks occur throughout the year, throughout the world. I'm not going to show you the ProMed Mail search for that, but you can take a look. I wanted to show this one because I find it fascinating. There was an outbreak of Staph aureus during a Olympiad event, which was a college science Olympiad in which students were coming together to strengthen their science knowledge and test what they know about science. And pork sandwiches were distributed during that event, and they were contaminated with Staph aureus, and it caused a huge outbreak of Staph aureus foodborne infections. There's a nice summary here. Again, it's not the replicating bacteria that causes the infection once it gets into the host. It's the toxin that was produced before the bacteria before ingestion, and those toxins are heat stable, and that is what causes the symptoms. Susceptibility, everyone is susceptible because the organism is a normal member of the microbiota, but intoxication results only when inoculated food is improperly stored or undercooked prior to consumption. Treatment is self-administered replacement of fluids and electrolytes, and it usually passes relatively quickly. Now, for all of these, I talked about diagnosis with the dichotomous key, but you can also use something called X-tag GPP. It's a little more expensive, which is why it's not used quite as often as growing the bacteria, but it's much faster than growing the bacteria and looking for metabolites, metabolitic differences. This is based on PCR, so the DNA is isolated. There's some PCR amplification of the nucleic acids, and then detection by bead hybridization. It's detected on these very expensive but highly efficient and fast machines. Okay, so that's it for diseases of the gastrointestinal system. Well, no, that's it for diseases of the intestine. Now we're going to talk about diseases of the liver. These are referred to as hepatitis, hepa referring to the liver and itis referring to inflammation. So this is inflammation of the liver. Liver is an important part of the digestive system. It produces blood clotting factors, it stores glucose and other nutrients, and it helps in digestion of lipids and removing waste from the blood. We talked about this a little bit a couple of lectures ago when we were discussing susceptibility of infections that chronic alcoholic patients have. And it's because in part due to a decreased immune response, but also in part due to inflammation of the liver. Now, a healthy liver should look nice, bright pink like this, but in hepatitis, the liver becomes enlarged. That's the swelling, and you can see that there are all these white parts here. This is a liver that has chronic hepatitis. This chronic hepatitis typically occurs months after the initial infection. So here we have a normal liver, you can get chronic hepatitis, and over decades, this can develop into cirrhosis, and eventually, if the patient has this increased inflammation of the liver, eventually this can lead to death over several decades. Hepatitis can also lead to jaundice, which is a yellowing of the skin and is often observed in the eye. So here you can see this eye is yellow and the skin is yellow, and that is due to an increase of bilirubin. Sorry, it's due to an increase of bilirubin in the urine, which is referred to as bilirubinemia. And so here you can see normal urine is a light color, and when bilirubin builds up in the urine, it can turns it very dark. And this is due to liver, a decreased liver function. Now, there are a number of viruses that can cause hepatitis, and this is a table contrasting hepatitis A viruses, hepatitis B virus infections, and hepatitis C virus infections. And what's interesting is that although all of these cause infections of the liver and can cause jaundice, they are all very different in the way that they are obtained and the outcome. So, for example, hepatitis A is often obtained or transmitted through ingestion of contaminated food and water or direct contact with an infected person, whereas hepatitis C is typically found in patients who have been injecting drugs or via an infected blood or organ transfer or unprotected sex. Hepatitis B can be obtained through contact with infected blood or body fluids, or mother-to-child transmission, or again, unprotected sex. So these come in through different transmission routes. And then they can cause very different diseases. So for example, in hepatitis A, it does not cause chronic liver disease and is rarely fatal, but hepatitis C can cause chronic infection and can lead to liver cancer or cirrhosis. Hepatitis B can cause a chronic infection that can lead to liver cancer or cirrhosis as well. Prevention, there is a vaccine for hepatitis A and hepatitis B. There is not a vaccine for hepatitis C. Now, in this particular chart and in the book, it refers to the fact that there is not a treatment for hepatitis C, but there is now a treatment for hepatitis C. It's very expensive, tens of thousands of dollars per treatment. For all of these, all A, B, and C, the symptoms are very similar. Fever, fatigue, loss of appetite, nausea, abdominal pain, joint pain, jaundice. So it's difficult to determine whether a patient has hepatitis A, B, or C just based on the symptoms. To identify these, you have to take a blood sample and then screen for the different types of viruses in the laboratory. And these most often are looking for antibodies against these different types of viruses. The other way to determine which type of virus a patient has is doing a PCR amplification of viral nucleic acids. So this is a comparison of Hep A, Hep B, and Hep C. The two that I haven't talked about yet are Hep D and Hep E. Now, what's really interesting about Hep D is that it requires simultaneous infection with Hep B to replicate. So if a patient just has Hep D, the virus can't replicate. It can only replicate in patients who also have a Hep B infection. We don't know entirely why this is true, but if a person has Hep B and then acquires Hep D, so they have Hep B and they acquire Hep D, then this can increase the severity of the infection as well. Hep E only causes a mild infection and does not result in chronic infections. Hepatitis affects people here in Alaska. The biggest one is hepatitis C. This is a mandatory reported infection, and so if somebody has hepatitis, the hospital is required to report that they have been infected. And you can see here that the number of cases of Hep A has decreased significantly, down to 0 in 2017. Hep B has mostly remained acute. Hep C has increased. So our incidence of Hep C has increased here in Alaska. And again, this is in part due to the fact that we have vaccines against A and B. We don't have a vaccine against Hep C. All right, the final thing that we're going to talk about is parasitic diseases. We're going to talk about two. The first one is Giardia intestinalis. This is a very cute little parasite that infects water and can be transmitted in water. Definitely don't drink water that looks like this. And if you're getting water from a natural stream, you really should filter it because you want to prevent yourself from getting Giardia. Although it's very cute, you can see that it has this, it looks like it has two eyes. Those are the parabasal bodies. Although it looks very cute, it can cause frothy diarrhea that's very smelly. It can also be transmitted in daycare facilities where infected kids get this cyst from the parasite on their hands, and then they play with the toy. Another kid pops that toy in their mouth, and then that cyst is ingested and can start the life cycle over again. So what is the life cycle? The parasite forms a cyst. That cyst is stable and resistant to the acid in the stomach, allowing the parasite to pass through the acidic stomach as a cyst, and it converts into a tropozoite in the small intestine. There it replicates by binary fission, so asexually, and then the tropozoites are able to attach to the intestinal cell lining via these suckers that the parasite has. And this is a side view of Giardia attached to the villi in the microvilli in the intestine. Now at this point, you can diagnose this by looking for oval cysts in the stool or that X-tag GPP test that I talked about earlier. This parasite uses that suction to attach to the intestinal lining, and it can leave little indentations where it's damaged the cells, and it leaves scars behind from this ventral adhesive disc. You can see that these Giardia can cover the intestinal cell lining, which makes it really hard to absorb nutrients, and so this can lead to malnutrition. Eventually, the tropozoites break off, leave the intestine, and are converted back into cysts, which are shed in the feces. Symptoms include severe, greasy, frothy, fatty diarrhea. So think about that when you're going through your cases or when you're going through your notes later. Think about what type of diarrhea is so associated with each of these. We talked about bloody diarrhea. We talked about rice water diarrhea. Now we're talking about frothy, greasy diarrhea. So be able to differentiate what type of infection a patient might have based on the type of diarrhea that they have. It also causes abdominal pain, flatus, which is basically gas, loss of appetite, poor absorption of nutrients, low-grade fever, and a rotten egg smell of hydrogen sulfide. That's very characteristic of a Giardia infection. This can infect both animals and humans, and so can be found in dogs as well. Treatment is time and oral rehydration. And then the last thing I'm going to talk about are tapeworms. Tapeworms are fascinating because they can grow to be up to 6 feet long. Tapeworms have segmented bodies, and so what we see here is a head that has a scolex. The scolex has hooks on the attachment organ that attaches to the intestinal lining. And then it has a long body. Each of these little things here are individual segments. These segments can develop mature eggs within the segments. And so when these segments break off, these eggs are released, resulting in more tapeworms. The tapeworm absorbs nutrients through its skin and its cuticle. And the two most common tapeworms in humans are Taenia saginata, which is found in beef, and Taenia solium, which is found in pork. These are usually asymptomatic infections, but they can cause nausea, abdominal pain, weight loss, diarrhea, and blockage of the intestines. When that happens, the worm has to be taken out very slowly to ensure that these little segments are not broken off, because if they are broken off and left in the intestine, new worms will emerge. Diagnosis is to observe eggs or proglottids in fecal samples. Treatment is a single dose of niclosamine or paracetamol. Prevention is thoroughly cooking or freezing the meat, because even just freezing the meat will kill the parasite. Also look at the meat and look for a mealy look, which is it's meat that kind of looks like it has worms in it, basically, and also good sewage treatment. There's a fascinating story about tapeworms in this podcast, or sorry, not in this podcast, in this blog. And in fact, if you are interested in parasites, I highly recommend the Creepy, Dreadful, Wonderful Parasites blog, which is which has weekly cases of different parasitic diseases. You can read through this on your own. It'll be in the lecture notes, and the answer to the infection will be here. Okay, the rest of this is all just information for you. So you can stop your lecture notes at number 67. These other slides just have information from the Morbidity and Mortality World Report, and it's all the different types of diseases, microbes that can cause diseases of the gastrointestinal system, their symptoms and treatments, etc. I just find it a really good resource. And then for studying, I've included all of the different questions that I would normally ask during class are at the very end of this, at the very end of the lecture notes. So that's it. Thank you for listening to the very end. Again, everybody's getting 4 points. You don't have to answer any questions on Blackboard for this particular lecture. And be safe, everyone, and hopefully your nerves and my nerves will recover soon.